The overall goal of this procedure is to accurately and reproducibly collect and measure mouse saliva following pilocarpine stimulation.
Measuring saliva production in rodents is a challenging procedure and is prone to high variability. The described method is simple and it can be repeated on the same animal in the multiple time points of the whole study.
This method can not only measure the volume of the saliva produced, it can also be used to collect the saliva to further analysis of bioactive molecules that may be secreted in saliva.
Ultimately, this will be of a great use when investigating Sjogren's syndrome, salivary gland fibrosis, or salivary gland regeneration in rodent model systems.
For this procedure, have pilocarpine hydrochloride aliquots prepared and frozen, not more than 3 months old. Do not refreeze any thawed aliquots. Begin with preparing 0.6 ml microfuge tubes. Carefully punch a small hole in the bottom of the tubes with a heated 18-gauge needle. Make 1 tube per mouse. Then, set these tubes into 2 ml tubes. Sterility is not a concern. Next, using aseptic methods, cut cylindrical absorbent swabs into 2 cm length. Then, cut each 2 cm piece diagonally to make 2 conically shaped swabs. Place 1 conical swab in each of the prepared microfuge tubes. Then, weight each 0.6 ml tube with its dry swab. Now, transfer the mice for the study into a new clean